Okay, so we are now being recorded. Okay, so this is our 7th video teleconference for BIOL 105 section YC. If we take a look at our calendar here, we are actually back in sync with the number of video teleconferences that we're supposed to be having. And that's because we rested for a while from video teleconferences for almost a month. Now we're back on track. Okay, so we are back on schedule. Um, we are also, in terms of lessons, we are also ending the lesson on chapter 8 today. Um, and there is a quiz from chapter 7 that will also be closing later at 8 p.m. So if you haven't taken that quiz yet after this video teleconference, you may do so. And you might be happy to know that in this video teleconference, we'll be reviewing the contents of chapter 7. So, which will also be part of the quiz, of course. Okay, so in a way, if you haven't taken the quiz yet and you are viewing this right now, you may get some information from here that will come out in the quiz later. Okay, so we are now in module 3, actually. We are done with the midterms. As for the midterm grades, I mentioned this to the other groups already, that your grades are not yet done. Okay, so I haven't submitted any of the grades yet for all of the sections, including the laboratory sections and genetics, the one that I'm handling. So please be patient and just keep calm and continue doing your best, no, in this after midterm season. So we will review chapter 7 this morning, and I want us all to be interactive, to share if we can share something in terms of the review. Okay, so without further ado, let's start. Let's review chapter 7, the interactions between cells and their environments. Before I show the first slide, it's important to remember that we are talking here about multicellular organisms. And when we're talking about cells and their environments, we're talking about cells in tissues. No? So particularly, most of the time, animal tissues. No? So later on in the end part of chapter 7, there's a part about plant tissues as well. Okay, so first, let's start on the surface of the cell itself. No? So the surface of the cell, of course, it's covered with a plasma membrane, as we can see here. Let me have my laser pointer. So here we have our plasma membrane, and then we have some structures that are extending through the plasma membrane, and then we have these green fibrous structures. So before we even talk about the ECM or the extracellular matrix, can anyone from the group tell us what we are seeing here, particularly these green structures?
Hello, sir.
Yes.
This is known as the glycocalyx or the cell coat. It is made of oligosaccharides, sir, and it mediates cell-to-environment interactions as well as cell-to-cell recognition and communication, sir.
All right. So everything that you've said about the glycocalyx, no, that's what is called, is correct. So even if I started with the statement that we will only talk about multicellular organisms, the glycocalyx can also be found actually in unicellular organisms, though in microbes, such as in bacteria. Excuse me. So in the video that I posted, I actually included there a colony of bacteria. And you can see that the colony is sort of mucoid, no, parang murag mucus. And the mucus is actually because of the secreted carbohydrates by the cells of the bacteria, which is also the glycocalyx. So it's used by bacteria particularly to attach onto things and to each other. So for example, if you haven't brushed your teeth for a day and you touch the surface of your tooth, no, they will feel slippery and almost mucus-like. No, there's a thin mucus-like layer. And that's not just because of your saliva. That's actually because of the bacteria that are starting to colonize your tooth. No, and they have glycocalyx, so that's why it feels a bit slimy. So that's an example of glycocalyx. In animal cells, of course, it covers them and it mediates in cell-to-cell interaction or cell-environment interaction. While in plant cells, I don't think we can find any glycocalyxes. All right, so now let's move on outwards away from the glycocalyx. Okay, now the extracellular matrix, particularly in animals. So in animals, there are two main groups of tissues that have this extracellular matrix. Now first, we have the epithelial tissue as shown here on top, and the connective tissue, the one here at the bottom. Okay, so can anyone from the group tell us the difference in the extracellular matrices between epithelial versus connective tissue? Anyone? I encourage you all to at least have a speaking part. Okay, because when you speak it, you learn.
Epithelial tissue, sir, it consists of, as you have seen in the picture, it is consists of closely packed cells, and underneath it is the its supporting structure, the basal lamina. While the connective tissue, it is as what I've seen in the picture, it consists of largely of extracellular matrix, and and its function is for cell receptors, sir.
Okay. So thank you. No? So what you've said was correct. You observed, no, that's good. You observed from the illustration that in the epithelial tissue, the cells are close together. No, they're sticking together and they are all attached to the basal lamina, which is the extracellular matrix for the epithelial tissue. While in the connective tissue, um, the cells are not really close together. In between the cells are the extracellular matrices, no, which you mentioned also helps them interact with each other and with the environment. So that's the difference in the ECM. No, we'll just call it ECM between epithelial tissue and connective tissue. So basal lamina, a thin sheet of of ECM is present in the epithelial tissue, while in connective tissues, the ECM acts like a filling, no, sort of like a cement in a wall of bricks. So the bricks are your cells, and then in between the bricks are the cement that would stick all the cells together. That's the ECM. Okay. Additional question from this illustration. Can anyone from the group tell us what these black structures are in the epithelial tissue and what these red structures are? You have here at the bottom and you also have here in between cells.
Hello, sir.
Yes.
For the black portions, those are um, the ones in between cells are the cell-to-cell adhesion sites. The ones between the basal lamina and the cells are the cell to also addition sites, sir. And then the red portions are cytoskeletal filaments, sir.
All right, so you are correct there. No? So specifically later on, we'll talk more about these. The ones that attach the cell onto the basal lamina are the hemidesmosomes. No? These are the hemidesmosomes. The ones that attach the cell to another cell, the one here at the bottom could be the desmosome, and the one here on top could be the adherence junction, if you can remember that. And between the desmosomes and hemidesmosomes, the fibers here, you mentioned the cytoskeletal fibers, are intermediate filaments. And in the adherence junctions, the fibers here are the actin filaments. Now, so just an overview. So thank you. Let's move on. So now the structure or the components of our extracellular matrix. Now we have five main molecules that are playing a role here. One of them, of course, is attached to the cell, the integrin. So let's first talk about the first four, the ones that are strictly in the extracellular matrix. Let's start with these. Can anyone from the group tell us what's shown in this slide?
Um, I think, sir, these are collagens. As and the one on the left side, specifically, is a type 1 collagen fiber, which is a structural fiber that is found in the corneas of our eyes. So, as you can observe here, they are so orderly that light is able to pass through the layer, and so that's why the cornea seems transparent. Not seems transparent. Cornea is transparent. So, and here on the right side, type 4 collagen, um, it's present in the basal lamina in the epithelial tissues, and they are arranged in a network-like structure.
All right. So thank you for elaborations. No, this is everything that you've said is correct. These are collagen fibers. No, um, thank you for that. But I have a follow up question. So anyone from the group can answer. What property of collagen makes it very useful in the extracellular matrix?
High tensile strength, sir.
All right, so they have high resistance to pulling forces, no, or tensile forces. They have high tensile strength, so high resistance to pulling forces. So in your skin, for example, if someone pinches you, your skin will just spring back to its original position because there are collagen fibers, no, on your in your epidermis and in your dermis as well. Okay, so that's very important for collagen. Okay, so that's it for collagen. Are there any additional information? The structure. Who can describe the structure of a collagen fiber? General
Uh,
structure, yes.
it is cancer. It is made of three alpha helices that sort of overlap each other, sir, and then so that it kind of forms a collagen fibril, sir.
All right. So the collagen fibril is actually these no. Okay. So thank you. So that's everything about collagen no, in a nutshell. So that might come out in the quiz later on if you haven't taken the quiz yet. So please read more about collagen. But everything that you've discussed and said are correct. Okay, so let's move on. Next, we have this molecule in the ECM. Anyone from the group tell us what this molecule is? and maybe its function and description.
This is cancer, proteoglycan, sir. And in this main red, lagi red sort of stalk, sir, it is made of hyaluronic acid. And then it also has these branching parts. And in the left part, um, the main branch is like, it's made of core protein. And it also has sulfates, sir. The longer ones are made of chondroitin sulfate, the shorter ones are keratin sulfate. And the sulfates are present so that it attracts water and it sort of makes a water padding around it, sir. So it has high compressional strength, sir.
Okay, so everything you've said is correct. So the name proteoglycan, it's very obvious what it's made of. It's made of protein, so we have some proteins here, and then glycan, carbohydrates. So the chondroitin sulfate, keratin sulfate, these are actually carbs, no? Also the hyaluronic acid, as you can see here. So that's why it's called the proteo, protein, and then glycan, carbohydrate. And as you mentioned, the presence of the sulfate ions here causes water to accumulate in between these carbs that we see here. And it acts like a padding, as you mentioned, so that a proteoglycan molecule has high resistance to compressional forces. So when proteoglycans and collagen fibrils are present in a connective tissue, the connective tissue will benefit from both of their characteristics, no? Just like in your skin. So your skin is not only stretch resistant, it's also compression resistant, no? So when you are being pinched, your skin will sort of um, resist the um, pushing force of the pinching because you have um, proteoglycans in your skin tissues. And when you pull your skin, it will just spring back to its original position again because of the presence of collagen in the tissues. All right. So next, we have this molecule. Please describe the structure, function, and the name of this molecule. Okay, go
It's
on. Sige,
fibronectin, sir.
correct. Okay. Can you describe it based on how it looks like?
It contains sites for collagen binding, for heparin binding, and integrin binding, sir. And as well as its integrin binding and integrin binding receptors contains RGD sequences.
All right. So everything no is actually shown in this figure. So fibronectins have collagen binding and integrin binding and even heparin binding domains. And particularly for the integrin binding domain, there is an RGD sequence. The name RGD comes from the first letter symbols of arginine, glycine, and aspartic acid, if I'm not mistaken. aspartate. So another important thing to note is that at the C terminus, the two polypeptides that make up a fibronectin are bound together in a disulfide link, no, disulfide linkage. So, if I may go back to the previous illustration here, you can see the fibronectin, no, the purple ones, they look like hair clips. You can see here in this portion, that's where the disulfide linkage is. And then this is the free end. And you can see this is the integrin, the green one, and it's binding to the fibronectin at the integrin binding domain. No? And the fibronectin may also bind to other molecules like collagen, as you see here, because there are also collagen binding domains in fibronectin. Before we move on, function. What's the function aside from binding to collagen, integrin, and other stuff in the ECM? In the development of multicellular organisms, particularly animals, what's the role of fibronectin?
It guides the migration of cells, sir.
Okay, so its presence is actually detected by integrin, no? Because there's an integrin binding domain, and integrin is found on the cell membrane, as shown in the previous illustration. So the cell that's migrating, as you mentioned, no? Because during development, cells need to move from one place to another in the embryo. Um, so the pathway is actually being marked by fibronectins, so that the integrin can actually detect that that's the correct pathway for the migrating cell. Okay, so without fibronectin, the cells during development will be in chaos. They will not know where to go, and the development will be abnormal in that case when there's no fibronectin. And then we have this structure. Can anyone from the group tell us what's shown here?
Hello, sir. This appears to be laminin, sir. Um, it is made of three polypeptide chains, which are like intertwined with each other, and it also exists within the basal lamina, sir, along with type 4 collagen, as shown in pink, sir.
Yes, all right. So this is actually the microstructure of the basal lamina, no? And the laminin that you were mentioning are these green cross-like molecules. And as you mentioned, they are made up of three subunits that are intertwined together, and they have collagen binding domains. These are type 4 collagen, as you mentioned. So if I'm going to go back again to the previous illustration, this one. This network is the type 4 collagen. Now, we cannot see the laminin in this particular picture, but this is actually the pink, no, in the illustration a while ago. And if I will go back some more, the basal lamina, this is the one that was shown a while ago, no, the pink and the green colors, this one. So it looks like it's as if you're looking at a piece of paper under the microscope. Then you see the fibers making up the paper. So parang ganon. The basal lamina is actually thin. It's like paper, no? It's thin. But when you look at it closely, you will see that it's made up of a network of type 4 collagen. and together with the laminins. Um, the laminins, just like the fibronectin, also help for guiding migrating cells during embryonic development, no? Because they are found on the basal lamina. So basically on the surface where the cells crawl or move on. All right. So those are the four main molecules that are exclusively in the ECM. Okay. So before we move on to the next one, the one that's attached to the cell, we have this illustration. No? So here you can see the basal lamina. Okay. And here we have the epithelial tissue here on top. Here at the bottom, we have the connective tissue. So can anyone from the group tell us what's happening to these pink cells?
Yes, sir. The pink cells are cancer cells, sir, and they are known to produce excessive amounts of matrix metalloproteinases or MMPs, which have a function in the breaking down of the integrity of the ECM, sir. They digest ECM components, um, so that's why cancer cells are able to move. Um, they are able to metastasize into the other organs, sir, because they can move easily in the ECM due to MMP, sir.
Okay, so that is correct. No? These are cancer cells that produce excessive amounts of matrix metalloproteinases. Those are the enzymes that can um, destroy the integrity, no, of ECM, as you mentioned. So in the video that I posted, I think I described how MMPs work in terms of renovating your house. When you renovate your home, you want to expand, you need to destroy some walls. Diba? To expand. So cells naturally contain MMPs so that if ever there's tissue damage or growth needs to happen, the cell will be able to use the MMPs to destroy the ECM so that it can divide some more and then there's growth to happen. But cancer cells produce excessive amounts and they do not use it the right way, the way it's supposed to be used. Instead, they use it to their advantage to travel into the other parts of the body so that they can establish new tumors, no, in a process called metastasis. So MMPs are enzymes, and enzymes need cofactors. Can anyone in the group tell us what the cofactors are? Examples of cofactors of MMPs.
Zinc and calcium, sir.
All right, so
Uh,
zinc,
yeah.
continue.
Ano, sir. So, yun, they need them for cofactors to, for them to work, and they are entirely dependent enzymes that also can digest various ECM components.
All right, that's correct. No, zinc and calcium ions, no? Ca2 plus and Zn1 plus. So cofactors are inorganic, di ba? Inorganic ang calcium and zinc that's needed by the enzyme so that the enzyme can do its work. We also have another type of helper for the enzyme we call the coenzyme, and those are the vitamins, and they are organic. No? So I just want to plug that in to differentiate cofactors from coenzymes. All right. So thank you. So we now at least understand this illustration. So here's the fifth component which is attached onto the cell membrane. So we can see the bilayer here in blue. And we can see the structure is a dimer composed of an alpha and a beta subunit. This is when they are inactive, they're bent and they're sort of together. But when they're active, they move apart a bit and both ends on the outside attach to certain ECM components like the collagen here, and also in the inside, um, like the talin here. So what do we call these molecules? The green ones.
Integrin, sir.
All right. Why are they called integrins?
I think, sir, because they integrate into the cell in the intercellular and at the same time at the extracellular, sir.
That is correct, no? So that is correct. And one thing also that comes into mind is that they are integral proteins, no? They span the whole width of the membrane bilayer. So integral proteins, and they integrate um, messages from the outside to the inside and vice versa. So that's correct. Integrins are important, particularly in what's happening in this illustration here. So here you have step 1, stage 2, stage 3, stage 4. Anyone from the group tell us what's happening with the cell?
They are in the process of focal adhesion, sir, they in which the surface of the cell anchors to the surface of the I think it's a petri dish.
Yes, that's correct. So if you put a cell on a surface, no, on a solid surface, the cell will actually, in time, hold on to the surface. So you can think of the integrin, this one, no? You can think of this, look at this. It's like a hand that's holding on to something. So when you put a cell on the petri dish, as you mentioned, this is glass, the integrins on this surface of the cell will hold onto the glass surface, and the cell will secure itself onto the glass so that it will stay put. Now, so you imagine yourself riding a roller coaster and you're about to do a turn, 360 turn vertically. You hold onto the railing so that you will not fall off, just like what's happening to the cell here. So the cell, what is it using? The integrins to hold onto the glass surface so that it will not be rolling about, right? So, and that's called, as you mentioned, focal adhesion. So here we can see the integrins are actually the ones that are really lighting up, no, in a focal adhesion. But not just the integrins, there are also actin filaments. So you can think of this as the integrin as the hand holding on, and the actin filament is the arm that's attached to the hand. Diba? So, without the actin filament, useless ang pag-hold on ni integrin because there's no force that will keep the cell from holding on. So, kailangan talaga may actin filament. Okay? So, that's focal adhesion. And here's another look. Here you have the surface of the glass or whatever surface. Then here you have the integrins. They look like the tube feet, no, of a starfish. No, if you've seen the tube feet of a starfish, they attach onto the glass and they are connected to the actin cytoskeleton, as you can see here in blue. That's focal adhesion. While the cells in the epithelial tissue hold on to the basal lamina using a hemidesmosome, no, as we see here. So can anyone describe to us the structure of a hemidesmosome or how it works? You may just use this illustration. Huh? You can describe already the structure.
Yes, sir. So the hemidesmosome involves this plaque containing protein, the yellow one, but instead of actin filaments, intermediate filaments of the excuse me, of the cytoskeleton is in used, sir. And also integrins are also involved, but they are attached to the laminins found in the basement membrane, sir.
All right, so thank you. No? So here you have the basement membrane with the blue type 4 collagen, and then you have here the cross-shaped laminins. Attached to that is the integrin. Integrin again, di ba? But this time, instead of being attached to actin filaments in focal adhesion, the integrins are attached to this plaque protein, plaque-containing protein, and instead of actin filaments, intermediate filaments are the ones that are anchoring the cell. So intermediate filaments are actually less flexible. So this means no, that the cell is really secure, strongly attached onto the ECM, basement membrane in this case. So I have also here four other cell-to-cell junctions. So anyone just choose a cell junction and then let's describe it, its structure and function.
The adherence junction, sir, by its name, it adheres one cell to another. It contains adherence and which connects to the actin filaments. Then, aside from adhering, its function is also to provide a pathway for cell signaling.
Okay. I think for the pathway, this is not the main junction. Okay, but everything that you've said aside from that, no, are correct. So, okay, that's correct. Next, another junction. So we're done with one.
For the desmosomes, sir, it also functions as cell-to-cell adherence, but instead of normal adherence, it uses desmoglein and desmocolin, which are attached to the um, in the inner membrane of the cell, sir. It is attached to placoglobin, also which is also attached to desmoplakin, and intermediate filaments are also involved in this, sir.
All right, that's correct. No, so the first two, adherence and desmosome, they are for cell-to-cell adherence. The only difference is in terms of location, adherence junction are usually higher, no, in this columnar cell nearer the surface, while the desmosome is usually located nearer the basal lamina. Now that's another difference. And of course, the other ones are very obvious. Actin filaments working here, and intermediate filaments working here, and also on their adherents that are involved now in the cell-to-cell adhesion. Right, so we're done with two. We have two more junctions. Anyone?
This tight junction, sir,
All right.
it's staple-like structures that are composed of the Claudine proteins. So they act as prevention, or they prevent the fluid from passing through. So it somehow prevents staples in between the cells that prevents extracellular fluid from going through.
All right, so that's correct now. So just to clarify, so if you can see my red pointer here, now this is where the extracellular fluid will be. So the extracellular fluid will not be able to go in between these two cells because there is a tight junction somewhere here. No? So that's what it prevents from moving through the extracellular fluid, as you mentioned. All right, so we're done with three. One more.
The gap junction, sir, it is composed of a integral protein called connexin, while its connexin is a subunit. It's the sixth connexin is called connexon, and its main function is for intercellular communication.
Yes. All right, so that's correct, no? So this is the part where one cell can communicate with adjacent cell because the connexins actually make a channel called the connexon, as you mentioned, which allows information from one cytoplasm to go into the adjacent cytoplasm. So again, no, some of us might be confused. Um, what we have just discussed are number one, the cell to surface adhesion, and we had two. First, we had the focal adhesion. Second, we have the hemidesmosome. Next will be the cell to cell, no cell to another cell, and there are four of them: adherence, desmosome, tight junction, and gap junctions. Okay. And then we have this illustration. No? This is actually a sequence of cells that are from an embryo. We can see here they are color coded. So there's something happening with these cells. And then these blue colored cells are sort of caving in, as we see here. They're separating themselves from the orange ones. And finally here, the blue cells are now completely separated and they're forming a neural tube. So can anyone from the group tell us what is behind this separation of the cells? I think someone is unmuted. Please check your mic, no? Unintentionally unmuted. Sige, that's fine. So, sige, so what is behind this mechanism? Why are the cells separating from each other?
So, Sir, so you can say in the first batch of cells in the upper left, um, these cells are expressing the same type of cadherence, sir, which um, uh, ano eh, like cells will only attract like cells, so obviously they are aligned. And in the second one, there is a blue portion, and these cells express different type of cadherence, sir. So they only recognize each other, which result to the third phase here, which they sort of kanang tawag ni? uh, they separate, sir, from the original sequence of cells, sir, eventually leading to this neural tube, sir, where they only recognize each other, sir, because of the different type of cadherin that they express.
Yes, so everything that you've said is correct. No, it's all about whether they are expressing the same coherence. No, so you will recognize, the cell will recognize similar cells as itself. So if we go back to the junctions, now what are we talking about the coherence? Remember a while ago when you answered, no, the adherence junction involves coherence. The desmosome also involves special type of coherence. So it's actually these types of junctions that are working, no, in this sequence. No, it's the adherence and then the desmosome junctions. Because of the differences in coherence, there is a separation of those junctions between the two types of cells, eventually leading to two groups of cells that we see here. So this is very important in development, no, of multicellular organisms. Otherwise, all of your cells will be the same and you will just be a big blob of similar looking cells. So no specialization in your tissues. So very important. Next, so this is in plant cells, of course. Okay, we can see here a channel between two plant cells, and here's a cell wall. Can anyone tell us what this structure is called? So it's drawn here. Okay, if you have the answer, you may speak because we only have around 7 minutes left.
I think, sir, this is the plasmodesmata.
Yes.
Uh,
Uh, function?
ah, um, this serves as channels between cell walls of two adjacent plant cells, sir. So the plasmodesmata allows the cells to communicate with each other. So it somehow is the same with the gap junction of the animal tissues, sir.
Okay, so thank you. I think you've said everything that I wanted to hear, including in my follow-up question. So, okay, plasmodesma is singular, plasmodesmata is plural. Okay, that's all that I can add. And then here, last of my slides is a special type of extracellular matrix found in plant cells. No, so this is the structure of the cell wall, and you can see here the cell wall is made of these big cellulose microfibrils that are arranged in parallel, and then perpendicular to that are the hemicellulose and the glycoproteins. And then we also have pectins in between the cellulose microfibrils. Okay, so what makes the cellulose microfibril? What enzyme makes the cellulose microfibril? It's already in this slide. Anyone? It's on the upper right. Nasa upper right ang enzyme. What's the name of the enzyme that makes the cellulose microfibril?
The cellulose synthesis, sir.
Okay, cellulose synthase. Okay, so they are made up of subunits, so they appear like a rose, no, a rosette. And then underneath, there's a cytoskeletal element, a microtubule that controls the location of these cellulose synthases. So that when they make the microfibrils also have certain directions. They're parallel. That's why they're parallel here. Okay. What about the hemicellulose, the glycoprotein, and the pectin? What makes them, or where are they from? Anyone? The answer is on the bottom right.
The Golgi apparatus, sir. The Golgi apparatus. So these other components are deposited from the inside to the outside.
All right, so that's correct now from the inside of the cell. So um, you can think of it this way. I don't know if I mentioned this in the recorded videos. The first thing that's made in the cell wall of plants is the cellulose microfibril made by the cellulose synthase on the cell membrane. You can think of the cellulose microfibril as the, in Bisaya pa, kabilya, no? Kabilya sa building. Now, before you construct the whole building wall, there needs to be the kabilya. And then later on, you put on the hollow blocks and then buhos, di ba? Ang term is buhos, buhusan ng semento. And then you can now have the wall. So it's like that when the plant makes the cell wall. First, the foundation, the cabillia, the cellulose microfibril. Then later on, once that's in place, from the inside of the plant, no, it will then add additional components like the hemicellulose, glycoproteins, and pectins. It's like making a wall, literally. Literally. Okay. So I just a reminder, quiz on chapter 7 is open until 8 p.m. today. So please take time to take the quiz. That's on the ECM, the one that we just discussed. And I hope everyone was able to participate in the group discussion for chapter 7. There is no group discussion yet for chapter 8. Just stay posted. I will just inform you through eLearn when it's ready for you to answer. So, we've ended on time. Are there any concerns with regards to this subject? I know many of you are part of genetics as well, which we will meet in 10 minutes. Pero Cellmol muna tayo. Any concerns with the lecture of Cellmol? Okay, so just additional words lang siguro, right? Because I thought of this, no? Notice how it's very important, not for you, no, as students, to really look at the illustrations in your book, no? Um, Many of us might be visual learners, so reading the text might be really very boring and very tiresome. But if you look at the illustration in context with what the text is saying, you will really understand also how things work. No, let your imagination do the animation. No, so, and it will be much more interesting to actually learn about these things. So, look at the illustrations and understand the illustrations found in your book as well. So that's my advice. Other concerns, questions? Wala na? Okay, so again, just message me if you have questions, all right? So I thank you all guys for joining in this video teleconference. The recording will be posted soon um, today, this morning, if you want to review on what we've talked about. So for those of you who are enrolled in BIOL 104 YC, I'll see you in 10 minutes. No, and you are now all dismissed.
Thank you, sir.
Thank you, sir.